be talking about neurons, synapses, and ion channels. So just as a reminder about membrane potentials, we have our resting potential at about -70 millivolts and our threshold at around -55. So the way that it all starts is we have our neurotransmitters binding to our dendrites over here. We have our graded potentials. But with each graded potential, we don't hit the threshold yet. But what we need is the sum of all the graded potentials eventually start to reach the threshold. So once we do reach this threshold, what happens? We get an action potential. And what happens when we get to the action potential? Our ion channels start to open. So these are voltage-gated channels. These sodium channels are voltage-gated channels, and at -55 millivolts, they're supposed to open. And what happens is sodium starts rushing into the cell, and we start getting positive, positive, positive inside the cell. So now we have rising rapidly. The membrane potential is rising rapidly. So as soon as we get to +40, these close, the sodium gated channels. And then at +40, the potassium channels start to open. And what happens then is sodium starts rushing out because of diffusion and because of voltage. Now, it starts going down. And the reason that it actually goes below resting potential is because the potassium channels actually take longer to close than the sodium channel, and so they don't close until too late, and so they let too much potassium go out, and then so it goes a little bit more negative. So I drew them a little longer here, longer than the sodium ones, just to help you remember that the potassium ones take longer to close. So what happens during this whole action potential? So what we have here is, in the soma, we have the rough endoplasmic reticulum, and that's where the neurotransmitters are made. And then in the Golgi body here, just like a regular cell, that's where vesicles are made, the neurotransmitters are made into vesicles. And so during the action potential, the neurotransmitters are rapidly brought across the cell, across the neuron, and it's brought to the terminal button, which are just the terminal ends of the neuron. So let's bring it to this one. And so how does it get into the synapse? We have exocytosis. Okay, and then it just binds with the postsynaptic cell.
Clinically, a slight imbalance of mediation of the ion channel may result in a lack or excess of neurotransmitter, making an individual more susceptible to neuropsychiatric disorders. Since the medications used to treat these illnesses also work in these ion channels, they are able to directly target the site of transmission and create balance by either exciting or inhibiting conduction.
The norepinephrine neurotransmitter and psychopathology. Alright, so here we have a brain. Some anatomy to know. The locus ceruleus, located here within the brainstem, is the major noradrenergic nucleus which is responsible for regulating arousal. The neurons within the LC contain norepinephrine. Axons project from the LC out to other parts of the brain, including the neocortex, hippocampus, cerebellum, and thalamus. When we say that norepinephrine is associated with arousal, we're talking about its signaling for the fight or flight response, for its impact on the sleep-wake cycles, can cause insomnia at high levels, it can affect emotions, it can increase anxiety, it can increase vigilance and contributes to focus, positively correlated with aggression. And then from things we've learned previously, we know that it is associated with the sympathetic nervous system signaling. So norepinephrine affects behavior through action on the cortical and subcortical brain regions. Signal disruption and or damage to the LC are associated with Parkinson's, schizophrenia, depression, and many other disorders. So decreased activity of norepinephrine can be associated with depression due to postsynaptic receptor downregulation, as well as ADHD. The decreased activity leads to problems with attention and focus. On the other hand, we have disorders associated with increases in norepinephrine activity. So this will include schizophrenia and the symptoms of it, and aggression in patients with personality disorder and mania. Manic episodes, such as in bipolar disorder, can also be associated with increased norepinephrine activity. Fluctuations in norepinephrine availability and activity can also contribute to anxiety and post-traumatic stress disorder.
Alright, so what is a neurotransmitter? They are molecules that transmit signals across a chemical synapse. So this is an axon terminal of the presynaptic neuron releasing neurotransmitters across the synapse. So the neurotransmitters are received by specific receptors on postsynaptic neurons, muscle cells, and gland cells. The 4 criteria for a neurotransmitter are that the molecule must be synthesized in the neuron, the molecule is present in the presynaptic neuron and is released on depolarization in physiologically significant amounts. When administered exogenously as a drug, the exogenous molecule mimics the effects of the endogenous neurotransmitter, and the molecule can be removed or deactivated by a mechanism in the neurons or synaptic cleft. So there are several types of neurotransmitters. There's acetylcholine, the biogenic amines, the catecholamines dopamine, norepinephrine, and epinephrine, the indolamines, serotonin, and histamine. Then you also have your amino acids, GABA, glycine, aspartate, and glutamate. Then you have your neuropeptides, including substance P, somatostatin, gastrin, oxytocin, the endorphins and enkephalins, the cholecystokinin, vasopressin, and luteinizing hormone releasing hormone. Then you have your purines, which are your adenosine and ATP, as well as gases and lipids, including cannabinoids, nitric oxide, and carbon monoxide. So for our purposes, we're just going to be focusing on acetylcholine, dopamine, norepinephrine, and serotonin for this video. Next, we'll be talking about dopamine. So dopamine is a catecholamine neurotransmitter in the brain that helps us regulate our emotional responses and our feelings of reward and pleasure. It also plays a role in how our brain controls our movements. Therefore, a deficiency in dopamine leads to Parkinson's disease, in which a person can no longer perform smooth, controlled movements, as well as depression. On the other hand, if there is too much dopamine or excess activation of dopamine receptors, then this can lead to psychosis and schizophrenia. So how does dopamine work in our brain? There are 2 main pathways that dopamine takes. So the first one is mesolimbic pathway, which goes from the ventral tegmental area to the nucleus accumbens, amygdala, hippocampus, and prefrontal cortex. This pathway activates our reward and pleasure centers in our brain. The second pathway that it takes is the nigrostriatal pathway, which goes from the substantia nigra in the striatum to the striatum, which helps control our motor function.
All right, so I'm going to talk to you a little bit about the neurotransmitter serotonin. So what we know is that serotonin, 90% of it, so the vast majority, is produced in our gastrointestinal tract. And we learned in our GI module that serotonin acts here to alter GI motility. And then a small percentage is produced in the central nervous system. And we know that based on research and how medications that affect serotonin levels show that increased serotonin generally shows improved moods, and deficient or low serotonin levels show decreased moods and clinical depression. So let's zoom in on our brain here to our neuron, and we can see how this kind of works. So our presynaptic neuron produces our serotonin here, and then when it acts generated, leaves that, enters the cleft. And we know this process following suit with the others, that there are serotonin-specific receptors on the postsynaptic neuron here, and they're going to bind to produce a response, which we think is going to be alterations in mood. All right, so then we have this leftover serotonin in our cleft here, which in a person without deficiency, we don't want just hanging around. So you have these reabsorption transporters here, whose job it is to take the excess or unused serotonin and move them back into the presynaptic neuron. And then that's where our SSRI come in as a treatment for clinical depression, because our SSRI comes in here and blocks our reabsorption transporter, leaving more serotonin in the cleft to act more often and for longer periods on these receptors, and hopefully act to improve mood and treat clinical depression. The other area for possible dysfunction or for treatment is through monoamine oxidase, which is an enzyme which breaks down our serotonin molecules into a byproduct. So monoamine oxidase inhibitors work by obviously stopping this process and leaving more available serotonin to leave the neuron into the cleft and then to bind to receptor. All right, so our last neurotransmitter is acetylcholine. And acetylcholine acts in both the peripheral nervous system and the central nervous system. In the peripheral nervous system, acetylcholine is going to cause muscle contraction. So it's going to act on muscle fibers and cause them to contract, producing movement. And in the central nervous system, acetylcholine is responsible for some pretty important things like memory and learning. So we have a light bulb here, as well as regulation of sleep. And it's also shown to work with movement coordination. Now, it's thought that low levels of acetylcholine may be related to Alzheimer's disease, and that high levels, there's some evidence to show that it may cause depression and anxiety symptoms. So let's look over at our neuron here and see how this works. So basically, acetylcholine is the product of acetyl coenzyme A and choline. And then choline acetyltransferase is the enzyme that facilitates this production. So we add those two together and we get our acetylcholine molecules here. We know with action potential, they enter the cleft, and after that, they're going to act on one of two or both neurotransmitters, and that's the receptors, and that's the nicotinic receptor and our muscarinic receptors. And the nicotinic receptors are located in the peripheral nervous system, while the muscarinic are actually located in both the central and peripheral nervous system. So obviously these molecules are going to come and bind here and then produce the actions based on where they're located, either muscle contraction or some of those autonomic like functions. And then it has to, the excess, just like with serotonin, has to re-enter our presynaptic neuron. However, with acetylcholine, it doesn't do this as the molecule acetylcholine. So we have this enzyme here, acetylcholinesterase, which breaks down our acetylcholine back into choline, which is then able to re-enter the presynaptic neuron and become available for more production of acetylcholine.